Module 7, the nervous system. Action potentials 1, the resting potential. I've gone over neuron and nerve structures. Now it's time to talk about how neurons actually send their signals. I already mentioned that the electrical signal that travels down the axon is called an action potential. Well, there are two types of cells that can detect or send that signal. This has to do with excitability. Excitability is the ability to undergo an action potential in response to a stimulus. Muscle cells and neurons are excitable cells. And though neurons are excitable cells, neuroglia aren't. When you learned about muscles, you learned that when a muscle cell comes in contact with neurotransmitters released at the synapse, it undergoes an action potential to signal the muscle cell to contract. Neurons also undergo action potentials when they receive a stimulus. An action potential is pretty much an electrical signal that involves ions, so this is both a chemical and electrical occurrence. And it involves a charge across a membrane. A membrane is a surface structure, so when it's disturbed, it will have a different electrical situation than any other cell part. This creates a measurable difference within a cell, and that's called a potential difference. A potential difference is a measure of the charge difference across the cell membrane. This difference is measured in volts, and the amount of charge is so small that the measurement is really in millivolts. When an axon is at rest, it has no action potentials running down it. Just outside the axon, which is just outside the cell's plasma membrane, the net charge is positive. Just inside the membrane, there is a net negative charge. Now, we aren't focusing on exactly what the individual charges are, but rather we're looking at the difference between the charges of two locations. That's the potential difference. So if you looked at this point on the axon and this point on the axon, you see that there is no difference. Both points are on the outside of the membrane and both have a positive charge. There's no potential difference between them. At rest, the entire outside surface of an axon has the same positive charge. Thus, there isn't any potential difference from one spot on the surface to another. Now, if you look at the outside of the membrane surface and compare it to just inside the membrane surface, you see that there is a potential difference of negative 85 millivolts. This is because the net charge on the inside of the membrane is negative. That results in a potential difference between those two locations. Well, when an axon is at rest, negative 85 millivolts is its resting potential. There's a potential difference in charge between the outside surface and the inside surface, and it's there when there is no action potential traveling down the axon. So at the resting potential, the inside of the neuron is negative. Now, there are actually two types of rest that an axon can have. If you have a large spring sitting on the floor, you can say it's completely at rest. But if you place a bowling ball on that spring, it's compressed, waiting to spring up when you remove the bowling ball. But in its compressed position, it is still at rest. It's just energized, ready to spring. A neuron's resting potential is like the energized spring. It had expended energy to get to that resting potential of negative on the inside and positive on the outside so that it's ready to go if and when an action potential comes. And at that loaded rest stage, the axon membrane is polarized with the positive charges on the outside separated from the negative charges on the inside. To help create this polarized situation, the axon has very large proteins trapped inside. The proteins were made by the cell, and they're too big to diffuse out. The only way they could be removed by the cells is via active transport. Another reason that there are negative charges inside the membrane is due to two positive ions, sodium and potassium. The cell undergoes active transport to send positive sodium ions outside the membrane. Active transport is necessary to do this because there are more sodium ions outside the membrane than there are inside. Additionally, positively charged potassium ions are actively transported into the cell because there are more potassium ions inside the cell than outside. These two ions and their placement around the membrane are a result of what we call a sodium-potassium exchange pump. It's called a pump because to move the sodium ions outside the membrane and the potassium ions inside requires energy. Now, you must be thinking that this pump exchange won't do much for the charges around the cell membrane because both ions are positive. But the sodium-potassium exchange pump can move 3 sodium ions out for every 2 potassium ions it moves inside. This results in more positive ions outside the membrane than inside. And the potassium ions tend to diffuse outside on their own, first because there are already more potassium ions inside the cell, and also because the cell membrane is more permeable to potassium ions than sodium ions. This creates even more positive charges outside. So to sum up, the cell membrane of the neuron uses active transport to move 3 sodium ions outside for every 2 potassium ions it transports inside, using the sodium-potassium exchange pump process. Also, some of the potassium ions diffuse back outside because their concentration is greater inside the cell than outside it, and the cell membrane is more permeable to potassium ions. Add to this the fact that there are very large negatively charged proteins that the cell has made, and these proteins are too large to diffuse outside. Therefore, at resting phase, there are more positive ions outside the cell than inside. This creates a negative resting potential in the axon of negative 85 millivolts because the inside of the cell is more negative than the outside of the cell. So you can see that though this is the resting stage of the axon, it requires energy to maintain this state, just like the energy it requires to compress a spring and hold it.